By the early 2000s, DNA testing had become the final authority for establishing legal identity. It was the genetic fingerprint that could settle disputes with nearly absolute certainty, providing a biological proof that was considered impossible to argue against. These tests operate on a single biological assumption, that every cell in your entire body contains the exact same genetic code. Whether a lab samples your hair, your blood, or your skin, they expect to see one uniform blueprint. This was the routine procedure Lydia Fairchild expected in 2002. As part of a standard child support case in Washington state, she provided DNA samples for herself and her children to confirm their relationship for the court records. The results should have been a formality. Instead, the laboratory reported that while the children matched their father, they did not match Lydia. According to the data, she was not their biological mother. Lydia knew she had carried and given birth to her children. But the state relied on the data. When an automated scientific system contradicts a person's word, the legal framework is built to trust the test and assume the person is committing fraud. The state's reaction was immediate. Suspecting Lydia of kidnapping or acting as an illegal surrogate, prosecutors moved to have her children removed from her care, turning an administrative filing into a fight for her family. The timing of the case created a unique opportunity for a controlled study. Lydia was currently pregnant with her third child. To eliminate any possibility of a mistake, the court ordered a witness to be fully present in the delivery room. They watched the birth firsthand, ensuring there was no chance of the baby being switched or the sample being tampered with. Blood was drawn from both Lydia and the newborn seconds after birth. When the lab ran the analysis, the result was the same. The DNA test claimed Lydia was not the mother of the child she had just delivered. This result removed the possibility of human error. It proved that the standard forensic model for DNA was missing something that allowed a mother and child to have different genetic profiles. Investigators ruled out lab mistakes and sample mix-ups. They confirmed the equipment was functioning as designed, yet the biological disconnect remained. A lawyer on the case discovered a medical report regarding a woman named Karen Keegan. Years earlier, Keegan's sons had failed to match her DNA during a kidney transplant evaluation, leading doctors to a rare biological condition. The condition is named after the chimera of Greek mythology, a single creature composed of the distinct parts of a lion, a goat, and a serpent. In medicine, a human chimera is an individual whose body contains two genetically distinct populations of cells. They are one person, but they carry two different sets of DNA. This meant the original tests weren't finding a stranger. They were simply looking at one genetic profile, while the children had inherited the other. This process begins at conception. Usually, when two separate eggs are fertilized by two separate sperm, the result is fraternal twins. But in rare instances, these two early embryos fuse together in the womb. Instead of growing into two people, they become one. As this single body grows, both sets of DNA multiply. This results in different organs and tissues developing with entirely different genetic blueprints. Investigators realized that while Lydia's blood carried one profile, her reproductive organs carried the other. They took a new sample, this time from her cervical cells, to test the DNA of the tissue that actually produced her children. This new sample was a perfect match. The DNA that Lydia passed to her children was present in her body, just not in her blood. Lydia was her own twin. The other woman the DNA tests were looking for was a second genetic identity residing inside her own tissues. With the chimerism confirmed, the court cleared Lydia of all charges. The threat to her family ended, and the case was closed with her legal maternal rights fully restored. The original laboratory tests didn't fail because the machines were broken. They failed because the biological model they were programmed to follow, the idea that every person has only one DNA profile, was incomplete. This case led to a shift in how genetic forensics are handled. Testing protocols were updated to acknowledge chimerism, ensuring that a single negative result from a blood sample is no longer treated as absolute proof of a non-relation. We rely on these tests to define who we are and who we are related to. But Lydia's case showed that biology does not always fit into a single, uniform category. It proved that a person can be genetically different from themselves, and that our tools are only as accurate as our understanding of the bodies they are measuring.